And a positive test would be described as? Zone of clearing. So when in doubt, just say ZOC, right? Okay? See, it's zone of clearing around the group. Zone of clearing, or ZOC. Okay. What kind? Lipase, because it's breaking down a lipid. Tributylin is a lipid. Remember, glycerol and fatty acids are the components of triglyceride. Okay, catalase test. Okay, some of you didn't see it, but some of you did, and we'll do it again later on. But this is a slide test, but you can also do it on a plate if you throw your plate away in the end, okay? And what you're going to do is you're looking for the enzyme catalase. How do you spell it? Cat-A-lase. Okay, but an S, not a C. Catalase. And what's the substrate? Hydrogen peroxide. What's its formula? H2O2. Okay? And what's the end product if it reacts and you get a positive test? Water and oxygen gas, O2. And always, when you show it, O2, use an arrow pointing up, which shows bubbles. Okay? And that's why you get the bubble that you see there. So, a positive test, it looks like Alka-Seltzer. Now, which gram-positive coxide is negative? Strep. Which one is positive? Staph. Now, if I change this test question, I might change it. Which one is positive and which one's negative? Make sure you know the right answer. Don't memorize. Okay? Or I'll mark you wrong. Because I'm evil. It's a guilt. Okay. Fermentation broth. We did that today. Okay? Right? What's the indicator? Phenol red. What color is phenol red when it's acid? Yellow. Fermentation produces acid end products and changes the indicator to a what color? Yellow color. Okay, that's a little redundant, but it helps. The small tube is called what?
Fermentation.
What's the small tube called?
Fermentation.
A fermentation or Durham tube. Okay? And what will it collect?
Gas.
It collects gas. And we'll talk about that today. When we get into lecture, we're going to go through glycolysis again. I want to show you fermentation. And fermentation sometimes produces CO2 gas. And that's what's being collected in this. Okay. All right, let me do this real quick while we're looking at this. Some of you didn't see a really good example. So I'm going to show you a really good example. If your gelatinase test didn't work very good, it's because you didn't inoculate it heavy enough. But if you'll look at this, look right here. See the two tubes? Which one's positive for gelatinase? The one at the bottom because it's liquid. Which one's negative? The one at the top because it's still solid. Okay, gelatin is a protein. If the organism grew, and you can see it grew there at the top, it's just digesting that gelatin like crazy, and so it's no longer solid. Okay, that's a positive gelatinase. Okay, so gelatin is a protein derived from animals. The test detects which enzyme? Gelatinase. A positive test is liquid after inoculation and incubation and refrigerating it in the refrigerator. So which two below is positive? B and C. Which one is negative? A. Okay, let's go to the next one. This was a starch plate. And Mary, did yours work? Did it work? Okay, good. Remember starch plate, you inoculate and incubate, then you add the reagent. Kind of like what we're doing today. We're inoculating and incubating, and we're going to add reagents next time after they've had a chance to grow. All right, the positive starch test, zone of clearing, okay? The name of the starch is amylose, which is the polymer of glucose, okay? And the enzyme we're testing for is amylase, A-S-E, amylose, amylase. The end product of starch hydrolysis is glucose. A monosaccharide with what formula? Duh. C6H12O6. What reagent is added after incubation to see the positive test in starch hydrolysis? Iodine. If you put Lugol's iodine, that's what we use, but iodine works. Okay? Now, Why? Iodine reacts with starch to produce a purple color. So if the starch is gone, you have a zone of clearing. Okay? Okay? So look up there. Is the E. coli positive or negative?
Negative.
Negative, because there's no zone of clearing. See, the one on the right is Bacillus subtilis, and it's definitely got a zone of clearing. Bacillus subtilis, if you notice, digests everything. It breaks down starch, it breaks down gelatin, breaks down casein. It's a soil organism. Soil organisms are tough. They get lots of enzymes because they have to eat what food is present.
Is it not amylase we can say that? Why? No amylase, right?
Oh, you can say, but no, why is it negative for starch? Yeah, it doesn't produce the enzyme. Okay, this one we're going to do next time. This one we'll do next time. This one we'll do next time. This one we'll do. I hate this test, the nitrate test. I dread it. Okay, MRVP. SIM we'll do next time. Decarboxylase we'll do next time. TSI we'll do next time. TSI, phenylalanine. Here's our DNase plate. Okay, DNase plates test for the presence of the enzyme. You missed this on me, DNase. What's the DNase plate? Okay. All right. The enzyme tested for is classified as enzyme group called blank because it breaks down blank. A nuclease because it breaks down a nucleic acid. What's the indicator? No. What's the indicator? What's the chemical dye that gives the color? Methyl green. Okay. MG, like the old British car that sometimes ran and sometimes didn't, because it had Lucas, Lord of Darkness, electronics. I knew it. Gallen High School. When hers was a Triumph. She let me drive it. All right. Never mind. Okay. Now, the pathogenic staphylococci are positive for DNase. What's MRSA stand for? Staph aureus. Methicillin-resistant Staph aureus. So if you don't walk out of my class knowing what MRSA is, I'll be mad at you. Okay? All right. MRSA. MRSA. Okay, we'll do this next time. Okay, come with your thinking brain on Thursday, because when I explain all of these, the MVIC tests, you know, you really have to put your brain on it to really get those things. All right. I'm done. Is everybody done? Got everything ready, put up?